Welcome to the first annual CCS Symposium. I hope everybody can hear me okay. I'm Lennie Woods, co-founder of the Clear Cell Sarcoma Foundation. We want to thank everybody for taking the time to be here. Most importantly, we'd like to thank those that are here and online who are fighting the good fight to find an effective treatment for clear cell sarcoma. So we're going to get started right away. We have several great speakers here. And I'm going to start with the next slide so I can see who's okay. We're going to hear from some patients. We're going to hear from Dr. Kevin Jones and Dr. Nobu and the xCures lady to talk about that, Denise, and here's some patient stories, I guess, if everybody's ready. Oh, first, let me do some housekeeping. There is a QR code here. If you want to scan it and you have any questions as you hear the speakers, you can post your question there, and they can also put it in the chat as well, right? Okay, you can put a question in the chat and we'll try to get to as many as we can. If you have it for a specific speaker, you can let us know that as well. And yeah, we'll get started. So some of the topics you're going to hear about are the current state of clear cell sarcoma, diagnosis and treatment. And I know some of you out there logged in could probably teach us some things. We hope that what comes out of today is the beginning of what we hope to be a long-term relationship moving forward in finding more effective treatments. But what we hope happens as well is an understanding of the purpose of why we need a consensus paper on the state of clear cell sarcoma. We want to drive the research to, prospectively. We have patients diagnosed every day, and we tend to find that we're getting the same treatments that we were getting decades ago. So we hope to change that and move forward. And, it's going to take the clinicians and the patients and families working together. So we'll talk a little bit about that and what's needed. Tissue from the patients, tissue to go to the researchers and the doctors, and how the relationship between patients, clinicians, families is so important. So next slide. So we're going to start the day off with Dr. Kevin Jones. He is from the Huntsman Cancer Institute. I'll let him tell you a little more about himself. Dr. Jones has got one of our clear cell sarcoma mouse models, and he's been an amazing champion for clear cell sarcoma and the Clear Cell Sarcoma Foundation for a few years now. So Dr. Jones, I'm going to pass it to you.
There, is that better? Okay. Okay, good morning, everyone. So I was asked to give a little bit of a state of clear cell sarcoma. And since clear cell sarcoma is a cancer, we're talking about biology, or the study of life. People have said that all biology is chemistry and that all chemistry is physics. But the difference is that in physics, we have things like Schrodinger's cat experiment, which is the idea in quantum mechanics that a cat in a box that is alive but has the means by which it would be killed, or poison, or some apparatus ready to kill it, until you open the box, you don't know whether it's alive or dead. So the cat could be in two different states. This is the idea, the theoretical types of experiments that they do in physics. And this is all very interesting. However, in biology, we just look in the box. And so we would find the cat either alive or dead, and that's just sort of what we get. Because in biology, we have two kinds of sort of knowledge that we gain. We look at things, or we make observations, or we prod things, or we test them with experiments. And so what I want to do quickly is go through kind of a brief history of clear cell sarcoma from the beginning, ways that the world of medicine has looked at clear cell sarcoma in different ways as the tools for looking have gotten better and better over the years, and how a few times they have prodded it with experiments or pushed on the cat to see if it was alive or dead. So how far have we come? Well, I just, downloaded this particular report or assessment from the NCI, the National Cancer Institute, just a couple days ago, giving the definition of clear cell sarcoma. It says soft tissue tumor begins in the tendons, and under microscope, it looks a lot like melanoma, and clear cell sarcoma of soft tissue usually occurs in the leg or arm, and it often spreads to nearby lymph nodes. It is most common in young adults. The challenge is that that sort of definition could have been made in the very first description of this, now, I guess, almost 80 years ago. So has there been any learning along the way? Well, one thing that was the first time it was reported was on 21 cases that a pathologist named Franz Enzinger reported on. He was at the Armed Forces Institute of Pathology. This was an institute. I don't even know if it exists anymore, but I assume it does. A lot of the sarcomas were defined by the AFIP back in the 50s and 60s. So Enzinger described 21 cases, and basically that was the first sort of real use of the term clear cell sarcoma. Another report came, 20 years later by another pathologist and some clinicians at UCLA. And again, they found that this was basically, adolescents and young adults, and it was a fairly aggressive cancer in that it often spread to the regional lymph nodes, and that about a little more than half of patients would die of it over the next sort of 5 years to a decade. One of the first opportunities to prod clear cell sarcoma or to test it came with the development of what is called the SUCCS1 cell line. What's fascinating about this is this was, again, 20 years after the disease was described, but it was also 20 years before we knew many of the molecular features of clear cell sarcoma. And yet this cell line is still the most used cell line in clear cell sarcoma research today. And it exhibits almost all of these, characteristic molecular features, which is kind of fascinating. I had not realized that this went back that far, but literally 20 years after this cell line was developed was before they even knew any of the sort of molecular things that we define clear cell by today. And those things primarily began in the 90s, where we started to notice that there was a chromosomal translocation that was usually observed in clear cell sarcoma. And the chromosomal translocations, this is between chromosomes number 12 and 22. So every cell in the human body has a group of chromosomes that include your entire genetic material. And two of these trade arms, basically. And at the position where they trade arms, they have a fusion between the beginning of one chromosome, the end of another, and vice versa on the other side. And so this was observed by looking at the chromosomes, which is a fairly complicated procedure in a bunch of, I'm not going to unmute my microphone, in a bunch of clear cell sarcoma samples. In order to get chromosomal spreads, what they call them, you have to have living cells that come out of a patient's tumor, and they have to be able to grow in a dish at least overnight. And so this was a prohibitively difficult sort of, procedure to put most tumors through. So this wasn't used a lot for diagnosis at the time, but this was the first recognition that this chromosomal translocation was identifiable and the first time was just a couple of cases. Okay, now I lost my. Okay. They found that this was beginning to be identified in most cases of clear cell sarcoma. So it almost began, and this is just a couple years later, to be kind of the defining feature of clear cell sarcoma. And then I put in this case just because this was the case identified clear cell sarcoma as arising not in an extremity or near the in the arm or leg or hand or foot, but actually in the intestines, and that is another common location where it begins. And we think of it as at least a very similar disease. It's not exactly the same, but it's a very similar disease when it happens in other places. It was a few years later where they actually identified the fusion gene that is created by this chromosomal translocation. So not only are they connecting, two arms of two different chromosomes, but you're creating a specific gene at that junction point where there's the beginning of one gene running into the end of another gene, and that it actually makes a protein that includes, apart from two different what we call parent proteins. So that was identified in the mid 90s as well. And then by a decade later, in the 2000s, this would have been the year I graduated from medical school. That was the first time that we started to define clear cell sarcoma by the identification of that fusion gene, because I told you the ability to detect chromosomal translocations is actually pretty difficult, but the ability to detect the gene at the single site, there were better tools developed by that point to identify that. So people started to apply these tools again to observe that in clear cell sarcomas defined by other features, by the way they look under a microscope, they were starting to find this particular gene expression. Okay. A few years later, this is just another look at a bunch of intestinal clear cell sarcomas, finding that they had the same feature. They had the same, gene, fusion gene expressed. This was, another almost decade later where the first two mouse models of clear cell sarcoma, and the reason why this is a prod instead of an observation, this is a test instead of an observation, is because really this was the first proof that that association that we had observed in clear cell sarcoma was actually capable of initiating clear cell sarcoma. That's from a scientific point of view, that's really important, because we can observe all we want, but until we sort of poke the cat and see whether it's alive or dead, this was the first attempt to do that. So we said, okay, we observed this fusion gene always present in clear cell sarcomas, but is it just a passenger or is it actually driving the cancer? So this was the first proof that it was driving the cancer, because we took out only that out of a human cancer and put that single gene, that fusion gene between these two genes that becomes one new gene, into a mouse, and two different groups did it, one group in Japan and our group in Utah. And it formed clear cell sarcomas. They looked the same. They had the same features. It was clear that that was the driving force behind this cancer. This is the largest cohort of patients that has been reported on, to my knowledge. And it's almost 500 patients. And you'd say, that's amazing for such a rare cancer to have a report of 489 patients across a decade in the National Cancer Database. The problem is that this is a little bit of garbage in, garbage out. What goes into the National Cancer Database is anytime anyone has used a code that is for clear cell sarcoma. The problem is that there are a lot of confusing codes. There's clear cell sarcoma of the kidney, which has nothing to do with clear cell sarcoma of the extremities and of the intestines and of some other places. And there are also clear cell carcinomas of the kidney, which are actually a very common type of cancer, that it is very easy for this just to be miscoded by, the thousands of community hospitals around that may have never seen an actual clear cell sarcoma, but have coded things as clear cell sarcoma, probably erroneously. So all of this has to be taken as, I would say, from the Midwest, a salt lick, not just a grain of salt. Those of you who have never seen, cattle in a stockyard or anything like that wouldn't know what a salt lick is, but a big block of salt. So there's a certain level of skepticism that we'd have with any of this data, but the basic observation is still pretty clear. About half of patients make it to 5 years, and you lose a few more in the next 5 years. This is a study from Japan, the sort of second biggest database study. And I would say that this is probably a much cleaner database. It's 122 patients over just 4 years, but it's still a database. It is a little bit more prospectively gathered database than the NCDB, but it's still a database, and it finds very similar findings. That their group of patients were a little bit older. Probably that just has to do with the way that they do their registry. Here is another attempt to make observations, again, molecular observations as our tools for looking into clear cell sarcoma changed. This was a paper we published a few years ago where we were able to do what's called copy number analysis on, and it was only a handful of human cases. This is looking for how many genes had extra copies and how many genes had removed copies in the typical cells. That was, again, an observation, but then we also were able to test a few, identified observations in the mouse model where we tested MID-F and MYC, two important oncogenes that are activated in clear cell sarcoma. We looked at how important they were to the formation of tumors. A very interesting paper was just a couple of years ago. And this, again, is an observation, but it's a very specific kind of observation where they were able to look into some clear cell sarcoma cell lines and try to understand exactly where this fusion protein, this EWS-ATF1, was actually located in terms of its regulation of lots of other genes. And so, again, it's an observational study, but a very powerful molecular observational study with new tools that we didn't have before. This is, I would say, a look and a prod. This is a group of sarcoma specialists around the world getting together and basically trying to sort of group their cases and how they had treated them. And they put together a total of 55 patients treated at 10 different centers and just tried to just sort of amalgamate a list of this is how it went with different types of treatments. And I think the main sort of take home was that clear cell sarcoma is very dangerous, and when you're needing to treat it with systemic treatments, it is often not going well. Another really interesting paper, and this is a paper that was very much supported by the Clear Cell Sarcoma Foundation, with Charles Keller and really a lot of people around the world donating samples to him. They did some more dense genomics. So again, a different tool for looking at clear cell sarcoma, the sequences of the genome. And they also developed a number of cell lines and tested a few drugs in those cell lines. And so this was, this I would say represents the best we've had so far in terms of the ability to look in a fairly large scale at a number of things and how they would work in clear cell sarcoma cell lines. And then this is a paper of ours from just last year where we used a very specific kind of tool to see how EWS-ATF1 functions in the cell or where it aligns across the mouse tumors. This is back to physics. So this is the Heisenberg uncertainty principle formula. And Heisenberg's uncertainty principle has really nothing to do with biology. But the principle is true in biology, and that sometimes when we're trying to prod things, when we're trying to look at things, our ability to see where they are changes where they are. And that's a problem. And so this mouse model that we developed was really an attempt to circumvent that. And so if you take a cell line and you say, I'm going to use this tool to find out where EWSR1 ATF1 is in the cell, What you don't know is you don't know if your tool is changing where it is. And that's a big problem. And that was actually a big problem with that paper I told you about a few papers ago, where they really did a nice job of trying to use the tools they had to understand where this fusion was, what other genes it was impacting. But the problem is that their method of looking certainly changed where it was. And so what we did here is we said, okay, we know we have a mouse that if we express EWS-ATF1, it will form a clear cell sarcoma. It looks like a clear cell sarcoma, smells like a clear cell sarcoma, walks like a clear, it's if it quacks like a duck, it's a duck. And then we added a tag to it. So we added a means of tracing it to it, and we put it in another mouse. And it turns out that it developed the same thing. So it could be changing where it was, but we knew that it wasn't changing the important places where it was. It wasn't changing its behavior, at least in its ability to make a cancer that looked like clear cell sarcoma. And so that we thought of as really a powerful tool to be able to a little bit circumvent Heisenberg's uncertainty principle. Again, don't tell any physicist you know that I said that. Okay, I am going to turn the time over to Nobu, who's going to share a little bit more detail.
I'm also big surgeon in Japan. Dr. Jones, I find the therapeutic target GLS as I defined with the mice, and also 613 plus HCQ combination clinical trial we should continue or not, we need to decide, and explore a new target of a cofactor, and also starting the liquid biopsy of sarcoma. So sarcoma is a very rare cancer, so to know the real result and to the more good way to do it, so international cooperation is needed, I think, so US and Japan. Yeah, so thank you for presentation today.
Okay, I have to say, wow. And and for our clear cell sarcoma family, I know we have some clinicians as well as some patients on Zoom. Kelly and Ann and Jill, Lisa, I think we should probably plan a deep dive for our clear cell sarcoma family on some of this work because it's incredible. And I wish that we had literally days for just your two talks. Thank you so much. This is incredible, exciting stuff. So we will now pass it on to a different, I want to sort of let our clear cell family know they've been hearing about this project, and many are still waiting in the queue. For those of you online, I know you're in the queue, you're ready to sign up. It's been an amazing hard work over these last several months, and we're getting there. We've made it through some hurdles and some barriers. For those of you who have consented, thank you. For those of you who are wanting to, we're going to get to you, but I'm going to pass this now. Which one of you first? Okay, Sarah Lewis from xCures, and then Hannah Hayes will speak after Sarah.
Can you hear me okay? Oh, wait. Good now? Okay. Lenny, you're making us follow that. That was incredible. So we are speaking a little bit about the project that we have been doing for Clear Cell Sarcoma Foundation and the incredible work that these ladies and the passion that they have. I'm very excited to be here today. I speak for Hannah and myself, saying thank you to the families and the patients that have already consented for us to be able to collect your data. And this slideshow is just a little bit about xCures and what we're doing with the data and how we're collecting it and using it. I thought I would start by just giving you a little bit of background on xCures. We started out with a company called Cancer Commons, and that company helps individual patients understand their cancer better by working with scientists and nurse navigators to better inform them of their cancer and trials and options that are out there for them. And where xCures came into play is we started and branched out from that for the ability and the necessity to collect medical records and the difficulty for cancer patients to have their medical records. And if you are a patient of any type of cancer, then you understand that you probably have, or you know, very rarely are just seen at one institution. You've gone to multiple institutions for second opinions and for consults. And what xCures wanted to do was make that easy for patients and be able to collect records from all different institutions easily for patients and for research. So we created a nationwide pan-cancer observational research protocol to be able to do that. And what that does is it allows us to be able to have the patient sign a HIPAA consent for us to be able to collect their records, and completely virtual. So the patient registers through our platform and then consents for us to be able to do that. And so basically they're consenting for us to be able to use their data for research purposes. Go next one. We have patients coming from multiple different channels. Important, bottom left is advocacy groups such as Clear Cell Sarcoma Foundation that have come to us, and that's just one of the routes that we obtain patient data from. I thought this slide was really interesting because we just have partnerships across the whole cancer ecosystem. So it just tells you a little bit how we're involved in lots of different projects. But top left corner, and I've got a couple slides at the end for you to refer back to, but it just speaks on some advocacy groups that we've worked with and what we've done for them. So I'll speak on that in just a few slides, but just wanted to share that what we use data for and how important clean data is for research purposes. So little diagram that shows how our platform works. So over the past 5 years, we've been creating a platform, like I mentioned, with the protocol to be able to consent patients. So they come to our platform, different routes. One of the routes being advocacy groups. We're able to consent them, and using the platform we've created, we are able to gather their medical records via HIEs, which is health information exchanges. So all medical records from all sites of care all over the United States. So we're able to query that usually within 5 to 10 minutes and be able to collect those records very fast and very simply for the patient. So all their medical records are in one place. And then you move into the processing, structuring of the data, which is another part of xCures model, where we've automated the data structuring process to structure, aggregate, annotate, clean data. And then metadata catalog is something that we are also developing, where you're able to search specific terms. So researchers can go in through a set database and search for a specific term that they're looking for. It just makes it easier for them. And then the fit for purpose data sets is something that Hannah's going to show you. That's specifically what you guys, have consented for your data to be collected for, and the data sets that will be used for research. So Hannah's going to share a little bit about the data that we've already received. We've had 14 that have consented, living and deceased, and we're very, very thankful for the families that are donating their loved ones' data for this purpose. The patients or the family members enroll. Sorry, Ann, you were good. Ann's doing my slides for me. So once they enroll, they sign up, and they give us consent to be able to query the HIE. And then we work with, or have been working, and we do work with the Clear Cell Sarcoma Foundation team to identify if there are missing sites in that data, so missing gaps in the data. And so we can actually, this is where the hands-on part comes in. We can actually request records from those sites directly, including, genomics reports, and then other diagnostic vendors that have a play in someone's care. And then we go in, our team goes in and we validate or verify and annotate the critical data elements. So when I say clean data set, I mean, without all the extra that come with it, what we want to see, what we want to pull out to be able to have a clear, concise data set. So conditions, medications, cancer medications specifically, any procedures, biomarkers. So the specific things that researchers are looking for, so they can look at those without having to look at all the extra sometimes I don't want to say junk, but extra things that come along with data. We also collect DICOM images from radiology departments. We de-identify those. So any of your records that you have consented for us to collect is de-identified. So no personal information is nothing's on there. So just for security purposes, I always like to tell patients that, because that makes you feel better about donating your data, knowing that everything's de-identified, so no personal information is ever shared with anyone for research purposes. And then we're able to export all the data with a standard data model and share with the CCSF team. And I'm gonna let Hannah share a little bit about what that looks like, and then I'll kind of wrap up last little bit.
Thanks, Sarah. And can you go back a slide? So I just wanted to touch on, hi, everybody. I actually have been with xCures for about a year, but have a background of about 20 years in clinical trials and clinical research. And so for those of you working in this space and studying sarcoma, it's the clean data. I understand passionately how important clean data is. And so on the point where we're validating and annotating the conditions, medications, procedures, biomarkers, those are, obviously very important points that for the research purposes. But I will say the beauty of what we do also is there is more data than just those points, right? Just those things we're annotating, because we are talking about real world data that we're pulling back. So you're also going to see vitals, labs, all kinds of things that are pulled back and extracted automatically as we pull the data in. So to me, that although the clean data is extremely important and that's what we're focused on, there may be opportunities to pull out things from, what comes back from the real world data. So I just wanted to touch on that quickly before going on to the next slide. Okay. So yes, again, thank you so much for all the patients who have signed up so far, and we're looking forward to enrolling as many as we can into this project. So far we have enrolled 14 patients, 10 living and 4 deceased. Of that data set, to what we've sent for analysis so far to the CCSF team is 8 patients. So we have a thorough longitudinal view of these 8 patients, and we intend to do the same thing as we move forward through the last 6 who signed up, and any of you who come in the future. And so we have also collected and de-identified 34 DICOM images for these patients. Just to give you an idea of the depth of information that we've received so far, just on the patients that the 8 patients, we have 1,458 medical records. So just on those patients, that is how much data we've gotten back so far. We've identified 62 conditions, 23 different types of pertinent medications to not just vitamins and different things. Those all exist, but 23 pertinent medications to anti-cancer medications, 14 different procedures, 24 biomarkers that we've analyzed, and across 132 encounters. So to me, I have a passion for data. Data is power. And I can just, see how this data set is growing and how it will grow in the future as we add to this, hopefully exponentially. And that little sample timeline, I know that's probably difficult to see on your screen, but what that represents, that's actually a view into our platform. Just looking at the cancer journey of a patient. Basically, you probably can't see very well on this little graphic unless you have a widescreen, but that's 2021. You can see there's a diagnosis there of clear cell sarcoma. And as it moves along, you can see the procedures that are performed, the medications that have been taken and what the start date is. You can also see, I guess there's, so conditions, procedures, all of that comes longitudinally up until it looks like 2023 in this particular timeline. So it kind of gives you a quick overview of exactly the journey of this particular patient and how it's been treated, how their sarcoma has been treated. And you can go to the next one. Okay, and then also a very powerful part of what we do, as Sarah touched on, is when we query the network, it brings back all the sites of care. So the patient doesn't have to indicate, oh, I was seen here, here, and here, go query these institutions. It brings back all the patient's records across all sites of care. So we've collected data from 90 sites of care, and you can see in this graphic some of the top 20 locations, location types that we've collected data from. So again, I know it's a little small, but you can see, you know, oncology, radiology, all of this analyzed based on the type of physician that has seen the doctor, or excuse me, seen the patient, and you know, brought back that information. So I just wanted to show this slide to give sort of an idea of the depth of the coverage that we receive. Okay, so this, I am going to go on record and say I am not fully qualified to talk about this slide, but I bet you all will find it very interesting. Here you can see these little hash marks represent our patients that have been seen. So you can see all of the patients have received the FISH testing. All but one have received NGS testing, and so on and so forth. And then as you go into the second part, you can see the alterations. So the EWSR1 ATF translocation that we were talking about earlier, you can see that on 100% of the patients represented here. Again, this is significant to me in what I know about the disease, but I'm assuming very significant to you and what the power it can have in driving research forward. Also, some other pertinent genes that we found in this review are listed here. And that is all I'm qualified to say on this slide. But hopefully you all can ascertain the significance of the possibilities here. And then this is what Sarah was referring to previously, some of the case studies that xCures has undergone in different cancer types. And I'll turn it back over to Sarah just to say a few words on these last couple slides.
Yeah, so just wanted to give you a little bit of a couple of instances where a couple of other advocacy groups we've worked with, DDRFA, which is a rare pediatric brain tumor. We're running an external control arm for them, collecting data to reduce randomization in clinical trials, which is extremely important. And then next, go to the next slide. Another type of rare cancer, fibrolamellar, working with them to collect data to be able to support their international tumor board with the records and summaries for research. So just a couple of, I know we're running out of time, so I just wanted to include those so you can go back and take a look at something that a couple of other rare tumors that xCures has been working with in the advocacy world. So just wanted to leave it there for you because I know you're going to share with everyone after this is over. So I just wanted to say thank you again to all of the patients and the families that have donated their data and what we're doing with it. And thank you for entrusting us with it. And really grateful to be a part of this project and be here today. I think I'm passing it on to you guys. Is that right? Yes. All right.
I want to say one thing real quick. Thank you, ladies, for that so much. Our team, Lisa, Ann, Jill, myself, have been working directly with Sarah and Hannah. And we have, this is really important to talk about and everyone in the room to know, because many of you in the room are included. Our clear cell sarcoma family in different parts of the country have been raising the funds for this specifically. And I don't even want to, I want to say all of your names, but you know who you are, these fundraisers you've been having for us. This is very affordable research for us to be doing. And we appreciate it. And I also have to say, for the families who answer my text at 10 o'clock at night and spend hours on the phone with me going through all the lists to make sure that we get it right. Thank you. And I want to say thank you, Hannah and Sarah, because they really see how the collaboration is important to us. It's important to get it right. We have seen a lot of data that's incorrect. Our family members, who I'm really speaking to right now, the CCS family worldwide, they've seen it. And these ladies here are committed. They don't just tell us, thank you, we've got this. They literally work directly hand in hand with all of us. So I had to say that and take the time. And we do have plenty of time because unfortunately Barbara's not here and I'm sure I'm going to flub her talk. So next, this kind of ties into the xCures project and everything that Dr. Jones and Dr. Nobu said. how the patients play such a role and how important it is to have everybody at the table and involved. So who's first? Lisa?
Thank you, Lenny. My name's Lisa Faulkner, and today I and Denise Reinke will be speaking to you on some patient advocacy and research opportunities. I just want to give a brief introduction of myself. I am a wife and mother of 3 boys, all grown. I have 3 daughters-in-law and 2 grandchildren. So that's a lot of fun. And I am also a nurse by background, over 41 years. I know I started when I was 2. Worked in the hospital since I was 14. So I've been around a little bit of medical things. I decided to get into nursing, and I've had such a great experience, lots of opportunities. My background has been in critical care, disaster medicine, some education and administrative opportunities. With that, lots of learning opportunities, and it's amazing what that brings to you when you get diagnosed with this kind of lovely cancer. I became a clear cell family member in 2020 instead of getting COVID like many of the rest of us. I decided I'd get this ultra rare cancer and started my journey, very adventuresome journey, as many of us have. Been initially misdiagnosed not once, but twice with melanoma. They just wanted to keep giving me that over and over again. It was quite confusing. My husband kept saying, "What? I don't understand." So we just kept asking for the tumor to be reassessed. And of course it was, but like we heard earlier, under the microscope, the stains seemed to look very similar, clear cell sarcoma and melanoma. So we continued the melanoma journey until my oncologist and surgeon had heard enough of my questions that they finally sent out for some genomic testing. And lo and behold, I have clear cell, and I had this little lump on my hip. And they were trying to tell me that I was in stage 4 melanoma, but it just didn't make sense because they couldn't find a primary. I swear they were just making all this up. And then it's just an inch away from my lymph node basin, and I didn't have anything in my lymph nodes. So we finally settled on clear cell sarcoma, and I found lots of opportunities for process improvement projects throughout the journey, from diagnosis through treatment. So I had some clear margins, luckily, and I also didn't have lymph node involvement, but they insisted on giving me radiation. And of course, there's not a lot of research out there, shocking. And so there aren't any real standards of care on radiation for clear margins, but they insisted that I needed that. I did have some kind of comment about, well, if this may be caused by radiation, why are we giving radiation? But they just kept on moving on, and we did radiation. I finally reached out to MD Anderson with my oncologist help and spoke to some gentleman who was really retired technically, but he actually created the melanoma sarcoma clinic at MD Anderson. And I had the honor and privilege to speak with him. So great guy. Anyway, he is now officially fully retired, so I've been abandoned. I'll find a new doc over there. But ironically, I was just a few days away from finishing my radiation when he told me that, oh, maybe you don't need radiation. I'm like, well, you're a little too late. And then he said, well, maybe you just need more. I'm like, well, let's pick one because I've got a few days left. So we just decided to finish what I was doing. And then it's like, okay, what do you do after that? Well, again, no real standards. There's no treatment. There's no cure, as we all know at this point. So I made a phone call to Lenny and here I am. So we've teamed up to do some patient advocacy and try to find some answers to some questions that we all have. And one of the major issues, of course, is being diagnosed or misdiagnosed. And a lot of us start out not in sarcoma centers, podiatrists, family docs, dermatologists. My dermatologist was like, oh, what's this? And some of us are lucky to find orthopedic docs. So how are we initially diagnosed? Well, usually there's some type of biopsy involved. And of course, the typical microscope immunohistochemical staining, and we get misdiagnosed with a melanoma, or maybe it's a lump, it's a fatty tumor, a lipoma. And so it just sits there and grows until it's eventually removed and hopefully diagnosed as clear cell. And then there's imaging. Well, what kind of imaging do we really get? And what's effective? You know, you've got this little lump. I just found a new lump a couple of months ago and had a new adventure with that. So what kind of imaging is really going to tell us what we need to know? Is it clear cell? That's what I need to know. Is it really clear cell, or is it 20 other things? And of course, when I made the phone call a couple months ago, my oncologist was out of town. And so I got the nurse practitioner and she says, oh, I think it's a lipoma. So many of us have been misdiagnosed with lipomas. And I looked at her and she was so sweet to see me right away. So I have to give her credit for that. And then I said to her, so how much do you know about clear cell sarcoma? You know, I wasn't being sarcastic, although I wanted to be. And she said, oh, just about as much, you know, anything my oncologist was sharing with her. I said, well, we kind of traveled down that road together. So we kind of know pretty much the same thing. And it may not be a lipoma. So she humored me and did an MRI, which the radiologist who read it somehow had this brilliant idea that it was under a scar tissue or under a incision. So it must just be scar tissue. So I called MD Anderson, and since I didn't have a doctor, they were gracious enough to try to get it to radiologists to read it. And so they just kind of went with the previous report. So it was under an incision, it must be scar tissue. Like, oh no, people, it wasn't under an incision. It's about 3 inches away. But so my husband and I had a trip to go on. We're 2 days into our drive and my oncologist gets back into town. She says, so are you interested in doing a PET scan? I'm like, sure. So we turned around, drove 2 more days back home to do a PET scan, to be told, oh, it's probably just scar tissue from a previous incision. People. So I went to see my surgeon the other day and she says, well, it could be scar tissue. We have done some digging around in that area. It is 3 inches from your incision, but we'll go with that for now. So needless to say, imaging is a challenge. And then how many of you have not had your lymph nodes assessed when you had your initial surgery? Luckily I did, but many of us have not. So that information is needed to help with more treatment and to remove those affected lymph nodes. And that leads us to another issue, treatment. There is no established effective treatment other than surgical removal, if that's possible. I mean, there are some medications that have had limited success with a few people, but unfortunately, since we're ultra rare and we don't really have the data nor the research yet, but we're getting there. It's been quite challenging. And then we need the pharma world to support us because there's a lot of money needed to get these trials done. And, you know, it's going to take a lot of time to develop a new drug. So can we test / repurpose medications? Unfortunately, a lot of us, you know, when we present to our doctors, have progressive disease. It's either in the lymph nodes or metastasized to a distant site. So how are you going to be treated? Well, unfortunately, since there's not a lot out there, they're going to go to something that's close to clear cell. Maybe they'll give you the Ewing's protocol, or maybe they'll decide on some other systemic treatments. Maybe you can become part of the Care Oncology clinic protocol. And maybe you've picked some dietary options. Maybe you're going to go keto, or maybe you're going to go vegan and hope that that helps. But unfortunately, we really don't know because we don't have a lot of data. And then where are you going to be treated? Are you going to be treated locally or at a sarcoma center? Well, how far away is the nearest sarcoma center? And, you know, there's so many different labels. Is it a sarcoma center? Is it a National Cancer Institute? Sarcoma center, what's the latest label? And how many patients do they really see that have clear cell sarcoma? You know, so you're really limited with your options. And then, you know, where's your support system? Hopefully you found us on the Clear Cell Sarcoma Family Facebook page. And then what are treatments that we're doing to ourselves that may even accelerate this disease? Again, we need to know what the data is looking like and to be informed on the overall survival, adverse effects, and the costs of doing some of these treatments that we have no data for. Luckily, we've partnered with xCures because data collection is a huge issue. Dr. Jones and Nobu have discussed some of the Japanese as well as the other data that's out there. We've been working with some SEER data that's in the US, and unfortunately that just doesn't give us the kind of data we need. And we need accurate, clean data. And how many of us have ever looked in our medical records to see what kind of data is misrepresented? I mean, every time I go in there, there's something new that's wrong. Just my latest experience with the whole imaging for this new little lump I have, you know, they get one thing in their mind and they just tend to stick with it. You know, the whole melanoma diagnosis. Well, you know, we had it independently evaluated somewhere else by a derm pathologist that we were so reassured that they would get it right. And, you know, they just, I don't know for sure, but I got this feeling that they read the prior report, look at what they're looking at, and they go, oh yeah, that must be it. So, I think I've spoken about the biggest opportunities that we can speak about in a few minutes or issues. And then Denise is going to talk to you about some opportunities with some of the things we've identified.
Thank you, Lisa. So I'm Denise Reinke, and I'm so pleased to be here today. Thanks so much for inviting me to participate in this day of really a deep dive into clear cell sarcoma. And by way of background, I'm a nurse practitioner, and I've spent 23 years focused in care of sarcoma patients. And while always maintaining a clinical care component, I was also part of a team to establish a sarcoma research consortium. It was a team of sarcoma laboratory and clinical researchers who recognized that really to make progress, they really wanted to work together, pool their intellect, pool their patient resources, and try to make progress and improve outcomes for patients with sarcoma. And that includes, it was broadly sarcoma, but was also inclusive of clear cell sarcoma. And while patients were patient advocates were always very interested and attended our meetings and participated, we really didn't have a real organizational structure to engage and bring that patient perspective to the research. And so about 5 years ago, we established a research advocacy council that we really fashioned after a model of the National Cancer Institute. They had established a research advocacy council of patient advocates who would come to the table and would give them their patient perspective into the work and goals of the National Cancer Institute. And so within the Sarcoma Research Consortium, we created a Sarcoma Research Advocacy Council. And that's really where I first met Lennie in person and really dragged her into this world of bringing the patient voice to research. And part of that initiative was bringing the patient advocacy community together to give them the tools and understanding of the research process and helping them to understand the important insights that they had to help us make progress. And as I think about if this meeting were to be held 25 years ago, it would likely be the research community that would be hosting this meeting and initiating it. And so I think it underscores the power and importance of the perspective of the patient that this meeting was initiated by this community. And so I think that we are really seeking to work together in a partnership, recognizing especially in uncommon cancers like sarcoma and specifically clear cell sarcoma, Nobody knows all of it themselves, and we have to work together. So Lisa clearly outlined for us the challenges that we face as a patient advocacy community from a standpoint of diagnosis or misdiagnosis, limited treatment options, kind of data that's kind of all over the place and not quite clearly co-located. And so I think that really challenges really give us opportunities and kind of lay the foundation of where do we have opportunities to make some progress, and how can we as a patient advocacy community really help move that forward. And so today I see there are three kind of key areas that we can focus on and really provide some input. And that would be in the collecting of data and the analyzing of data, and then sharing the findings. Really, because, and we keep talking about rarity, and I just to kind of define terms, if we go back to what the National Cancer Institute kind of defines as a rare cancer, that's a cancer that occurs in 15 of 100,000 people. That's their definition of rare. And then they have another subset of ultra rare, which is 1 in a million. And clear cell sarcoma is kind of put in that ultra rare category. And while some people may kind of quibble about what those numbers actually are for defining rare or ultra rare, I figured we'd kind of put a stake along with the National Cancer Institute. But it really does say that when you have a really uncommon situation like clear cell sarcoma, understanding the natural history of it, how does it develop? How has it been treated? What were the outcomes of those treatments? It's hard to get a sense. No one clinician has ever seen enough patients to get a gestalt of what is this disease and the trajectory. And so as a patient and family community, you have valuable information about what that journey is, how it was diagnosed. And so, Clear Cell Sarcoma Foundation really began this work with their Survey Monkey of gathering data. And that was just the very beginning nidus. Now we've heard about the xCures initiative. And we know that there's a third initiative that's currently ongoing as well within the FDA, where it's got a little bit of a different focus, but they're really looking to collect information about treatments that have been used for rare cancers. And whether it's an FDA-approved treatment or not, whether it's use of other substances, whether it's cannabis or nutraceuticals, they've selected sarcoma specifically as the cancer that they want to gather that information. So, here we have regulators who are saying, we want to better understand, are there treatments out there that are being utilized that maybe are effective, that aren't really already approved? Maybe there are treatments that are not effective or maybe even causing harm. So, it's kind of an unprecedented time as a patient community to be able to provide forth information. People are asking us for it, and we need to kind of step up and participate. And so we really do have that opportunity to share and give access to information. And then once we gather that, then we need to analyze that and say, what are we learning from this information? Are there some trends? Have there been delays in diagnosis? What's been going on there? Are there some opportunities that we can try to get people to where they need to be, or say, goodness, molecular, testing needs to be done, or what standards can we help after we look at the data to begin to generate some questions for future research. Now, we know we've talked a bit today about that, that there can be errors like in the medical records, or even in our own recollection. Sometimes we think we're going to remember all the details, but then when we kind of get to putting it down, we don't always remember all those details. So we just have to recognize that some of the data it's not going to be perfect. It's not going to be absolutely, 100% correct, but this is a starting point. And this will give us an opportunity for us to begin to analyze and say, well, what do we know, and where are the gaps, and where are the opportunities? And so then once we have this, at the mass sum of this data, then it's going to be really important for us to share it. And the patient community can be a part of it. There really have been several recent examples where patient advocates and researchers have published. So getting sometimes we think that publication is only something that, the super smart researchers like we have here in the room with us and the academics, but we as patient advocates can also publish this information, and it, in the form of whether it's consensus papers, or there's, what's the current state of diagnosis and care, or how do we jointly work together to set priorities, bringing all the views and perspectives together. And so getting information as published is so important, because it really gets it broadly out there and in the public domain that other people can see it. I think we also need to think about sharing the data with groups that establish guidelines. We've talked briefly, and I think we're going to talk more later, about the National Comprehensive Cancer Network. They're a group that has established evidence-based guidelines for the treatment of many different kinds of cancers, including sarcoma. And so, there's not much in there at this juncture for clear cell sarcoma. So do we have an opportunity that with the data that we gather to be informing the people that are putting guidelines together? Again, when things get published, then they become more broadly available and people can use it to no matter where patients are being seen, because we know not everybody gets to a major cancer center. And so having information available that community physicians can access is going to be important as well. And then also as patient advocates, we have an opportunity to share with the sarcoma research community. And we have a great example, that the abstract that was submitted from Clear Cell Sarcoma Foundation that's been accepted for a poster presentation at the upcoming Connective Tissue Oncology Society meeting, which is the international group of sarcoma experts. And a poster session is that there's like a big poster area about like 4 foot by 6 foot, where you put your information up and you have an opportunity as the researchers walk through this hall of posters to talk with them and exchange and share ideas. So we have an opportunity to share this data that we have. And then lastly, I think we need to remember to continue to keep the clear cell sarcoma patient community through the Facebook network and the newsletter and the website, so that you can continue to work collectively together and understand the status and the opportunities for sharing information. So I think today really is just such an incredible opportunity for us to work together, researchers, patient advocates, so that people facing clear cell sarcoma today and the people that will come in the door tomorrow and hear that they have clear cell sarcoma will have opportunities for new and better treatments and hopefully better outcomes. So thank you for having me here today.
Thank you. Thank you, Denise. And thank you, Lisa. Yes. So Lisa didn't say that when she called me originally, she wanted to talk about these NCCN guidelines that were so lacking. And I said, and who is this? And she told me who she was and her background in nursing. And I said, well, welcome to the team. Now, I don't want to scare any of our other CCS family members away, but we're waiting for you too when you're ready. So thank you. Thank you so much, ladies. And that ties into what we're going to discuss now. So Barbara Van Hare, who some know, here, everybody I think knows Barbara. Barbara unfortunately got sick and couldn't make it. And I can't even imagine filling her shoes. However, I've worked with Barbara for a while now. So I'm going to go through her presentation. Oh, sorry. I'm on this one using a different computer. Sorry. Here I can actually, you're going to go through the slides, right, Ann? Okay. So go ahead and I will start, Barbara. All right. So Barbara Van Hare is, and I try to remember how I met her as well. I know we have these funny stories. Like Denise said, she drugged me into the advocacy world. I'm so thankful that she did, but boy, I didn't know anything at that time. And Barbara is kind of the same way. And Barbara was working with some other groups that I really admire, other sarcoma groups, the Desmoid group and the EHE and Lyomayo. I've become friends with those ladies and Ewings as well. And what I've learned, what Barbara taught me was it's really hard. We talk about not having enough research and enough data, but what you need are research materials. And research materials, you don't think about this when you're just a patient walking around until you get thrown into the world and are trying to understand, how do I get information? How do I learn what's going to treat my clear cell sarcoma cancer? And you start understanding why that is that we don't have so much. And it really, if you look at our website, Dr. Jones had made us some videos a while back, and he talks about that and how patients need to get together and consolidate their data. Well, also we need tumor tissue, just like Dr. Jones and Dr. Nobu's talk. Those mice don't just come out of thin air. They had to come from a patient donating their tissue or a surgical tissue. And even then it's not as so we need lots of tissue, and being a ultra rare cancer, that's hard to do. So Barbara was connected with an organization a few years ago who was doing some tissue collection to be put on, and I'm going to mess this up. I know, Barbara, I'm so sorry, but who was doing some work at Broad Institute, Dr. Jesse Boehm, who Denny and I to have met early on in the foundation. He's an amazing scientist, working to create research materials to be put on the open work globally for scientists and researchers to be able to purchase at affordable prices. And they weren't just stuck in an institution somewhere and one research. So there was a grant that Dr. Boehm had received from the Department of Defense to help do this. And we are so blessed and thankful that he included us on that grant. So that's how we're able to get this work done. And the hardest part is just getting the word out there to the clear cell sarcoma family, and then getting the patients before surgery, because the tissue must be from, it can't be from a biopsy. It has to be from a surgery, and they only take what they need, and it's super easy, and it doesn't cost anything. In fact, I'm going to put her on the spot when Ann talks, if she wants to give a little bit, because she was one of the patients who was donated her tissue, and I think she can talk to anybody about it. But okay, so I'm going to go through Barbara's slides here, and if you can see them, we will also put the pattern.org. It's a link where you can consent. We'll put it on the Facebook Clear Cell Family page again. Ann's really good about sharing that. Super easy to fill out. There's a drop-down box where you pick clear cell sarcoma. If you know anybody who's got any of the different rare cancers, feel free to share it with them. They're collecting all types of tissue. Also, you can contact me or reach out to us through our website or the clear cell sarcoma social media pages. And Barbara, don't get mad at me for this, but she always answers the call. I can connect you directly with her. We don't ever want this to be a burden on the patient. We want it to be simple and easy, and you to feel like you have been heard and are understood. A kit goes out to your surgeon, to the hospital where your surgery is going to be. She doesn't like to have 12 hours notice, but we have been able to get it done in less than 24. She will do everything in her power to get that tissue and to get it to the hands of the scientists to create the research materials. I'm sort of winging it here, but I see she's got these great slides. Sorry. Y'all tell me if I missed something important. It does have to be set up before surgery. We get a lot of patients, and trust me, I know what it's like to be in the storm, and you can barely think about if you've eaten in the last 2 days, much less worrying about something like this. But we do, unfortunately, get a lot of calls from patients after surgery when they've had a minute to breathe, wanting to donate, and that is too late. If you have a loved one or a caregiver working with you, they can do consenting. Like I said, I can connect you by email. Barbara makes it as easy as you can imagine. And again, I want to reiterate, it doesn't cost you anything, and there's nothing extra that you have to do except for the consent. We just can't tell a hospital that you want to give your tissue and they say, okay. There are some hurdles that Barbara's an expert at. So we've got you, as you see, you can see the slides and you're going to get a copy of this, but it truly is something. Tissue, And I want to also go back to the xCures. You're consenting. How many do we have in the queue? I want to let everybody know. 30, 25 in the queue and 14 already in the xCures. So that's a pretty decent amount of patients. And we're not even pushing hard right now to get the information out because we're still sort of figuring out the kinks, which we're getting there. And that's just US based. And right, for our international family, they know, just hang in there because we will be able to take yours as well. We've got a big group ready to sign up. So just as important though as this tissue collection is your health record data. It all goes together for the scientists and the researchers and the doctors here to be able to make those decisions and those connections and what's working for better and a more effective treatments, longer lasting effective treatments though. So I think I've covered it all, right? Denise, did I do okay? Barbara, get better. We miss you. All right. So next we are going to hear from the, Jason Miller's family. This is an incredible story. And this is one of those families who I'm so humbled every time when you guys answer my 10 o'clock text and spend so much time on the phone with me, but it means a lot to me and to the other families. So I'm going to hand it over to you guys. Thank you.
Thank you. Good morning. My name is Amber Miller. I am honored to be here with my friend and sister-in-law, Kim Butler, to share about our sarcoma warrior and hero, Jason Miller. Jason and I spent 21 years together, 11 years married, 10 years as parents, and 4.5 months fighting for just one more normal day together as we fought the beast of clear cell sarcoma. Before we share about his battle, it is important to us that you know a little bit about Jason, as cancer and clear cell sarcoma did not define who he was. Jason was an amazing son, brother, husband, friend, colleague, manager, chef, believer, and most importantly, father. We were blessed with a 10-year-old son named Brody and a 6-year-old daughter named Cassidy, and he would have done anything in the world for them. He proved his love, strength, and fight for them as he pushed hard to take every last step he could until his body couldn't handle it. He taught all of us many things in life, but he most importantly taught all of us to never give up and to take one day at a time. We vacationed as often as we could and enjoyed spending time together as a family. He worked for an electrical distributor for 17 years and was very dedicated to learning and working his way from bottom to the top, and became very successful as the sales manager of CED&D. As his boss and best friend Chad said at his funeral, Jason wasn't successful because he was a good salesman. He was successful because he was a good human. Another important thing to note about Jason, he was a bit stubborn about going to the doctor. So on January 9th, 2023, when Jason called me and he said he was on his way to the doctor's office with fear in his voice, I was confused. The Minute Clinic, you don't have a family doctor. Lesson number 1. It is important to establish a relationship with a family doctor, especially as you age. Jason let me know that he called his family doctor's office to let them know that he felt a lump on his neck. And since his doctor retired 7 years prior, he was seeing a nurse practitioner. We had experienced inflamed lymph nodes due to colds and viruses, so I had hoped that they would send him home after another quick visit. But he said this time felt very different. And unfortunately, he was right. That doctor's visit led to an ultrasound, biopsy, scans after scans. And on Friday, February 3rd, 2023, we were informed that Jason had metastatic melanoma from the online portal. The nurse practitioner finally called Jason Friday evening before their office closed, and he read the results to him, but he could not answer a single one of his questions. He said to call back on Monday to ask for just one of the doctors. We cried, prayed, and worried all weekend. Metastatic melanoma did not make sense to us because Jason didn't have a single spot on his body, just a tiny bump in his neck. We called a good friend of ours that weekend who happened to be a dermatologist. Thankfully, she scheduled Jason for an appointment in their office on Monday with her partner, and she also recommended that we ask for a second opinion from a different pathologist. Lesson number 2, it is always important to get a second opinion. I had also called a local oncologist office that Kim recommended for melanoma, and they were able to sneak us in that very same day. He looked at Jason's biopsy results and recommended treatment as soon as possible. He scheduled Jason for a double immunotherapy appointment, and per our friend's advice, we pushed for a second opinion from a different pathologist just to be sure. Dr. Badano felt certain that the pathology report was correct with the metastatic melanoma diagnosis. However, he made a phone call while we were in the room with him and requested a second opinion on the pathology. On February 17th, 2023, we went to the hospital for what we thought would be Jason's first double immunotherapy treatment. But after several hours of waiting in the treatment room, our roller coaster was only beginning. Dr. Badano FaceTimed Jason from another hospital and let us know via an iPad that he received the results from the other pathologist, and this is where our clear cell sarcoma journey began.
Jason's battle was fast and hard with no time to even process all of the different things that were happening. Lesson number 3 that immensely helped during Jason's journey was having a healthcare advocate outside of the patient and the immediate family member to hear, advocate for the patient's needs, and offer support. I consider this to have been the most important job I have ever had in my life as his sister. Jason and Amber graciously allowed me to be a part of this journey and to be that advocate for Jason. From advocating for his medication to be delivered on time, to asking questions of the doctors, and just an extra set of ears to hear what the treatment team is advising is so very important. Due to the location of his tumor and the rapid growth he was experiencing, the struggle to manage the pain was next to impossible. This resulted in constant hospital admissions and readmittance with what felt like what end was very often every few days. The original plan of double immunotherapy was canceled with the confirmed diagnosis of clear cell, and it was determined that Jason's best option was to undergo 30 rounds of radiation to the mass on his neck. Due to the size of his tumor and the areas that it impacted, he was not a surgical candidate, and radiation was our hope to get it to shrink. Just before starting radiation, his oncologist had a change of plans and made the recommendation to undergo double immunotherapy based on a recommendation from the tumor board. Jason underwent 2 rounds of Urivo and Opdivo, with one treatment being just before radiation started and the second one being halfway through his radiation. While we felt like we didn't have time to waste and we had no choice but to move with the recommendation as they were given, it is so valuable to search out that second and third opinion, just like Amber mentioned. This gives you the ability to gain insight and advice from other doctors and subject matter experts in the field before making any decisions. On April 5th, 2023, Jason completed 30 rounds of radiation with hopes of being able to have surgery to remove the mass. Jason's tumor unfortunately had very little response to the radiation and the size barely decreased. After meeting with 2 surgeons, it was determined that the tumor could not be removed. Shortly after completing the radiation and immunotherapy, Jason's cancer took off and went from being isolated in his neck to his abdomen, his hip, and from there it spread all over. During this time period of radiation, double immunotherapy, hospital admittance, and hydration treatment, we did not know that Jason had the EWSR1 ATF1 mutation. The 4th lesson that is so important is to take the time and request that the samples be sent for additional testing. After fighting and pleading Jason's original biopsy, tissue blocks were sent to Tempest for additional genomic testing, which confirmed Jason's mutation and matched us with the local trial that Jason could have explored had we known about it a little earlier. It would have helped us to make a decision when it came to treatment options being offered, as we had the full genomic information of Jason's tumor. The last thing that I want to talk about is lesson number 5, and that is finding and sharing resources like the Clear Cell Sarcoma Foundation with others. We learned quickly that no one knew much about this cancer, and while we felt like we were on an island all alone, there are actually others out there, but we didn't know how to find them. After Lenny found our GoFundMe page and reached out for the first time, we were connected to someone who could offer support and guidance on questions to ask. I will never forget our first phone call in the hallway of the hospital, sharing just how bad Jason had gotten and how quickly. Having someone on the other end of the phone who could offer questions to ask, offer information, and just to be there and understand the fight you were in was such a feeling of comfort.
On May 18th, 2020, 129 days after his first doctor, Jason entered. He fought such a good fight. That Jason his patient. It was an aggressive cancer and rare cancer. No one, why wouldn't they want it for research? This is another hard lesson learned, and a lesson that we hope that you can all learn from. We were told that it was too, nothing else we could do. Hearing this news was one of the hardest moments for us, and something that will stick with me for the rest of my life. Since day one of Jason's diagnosis, we worked incredibly hard to find every treatment option. Jason's cancer to be researched. He was treated in 3 hospitals and surgeons. No one ever asked about donating his tumors. No one. That is a feeling that I hope that no patient or family ever has to experience. Here is the most important lesson we now know. And if you are a clear cell sarcoma patient who will be having surgery, it is imperative that you reach out to the Clear Cell Sarcoma Foundation to coordinate how to donate tissue. Do not wait for the surgeons and care team to ask, as it likely will not happen. We cannot stress how important that this is for the clear cell sarcoma community.
We must support and we must fight for the treatment options that make sense and are backed by true studies on clear cell sarcoma instead of relying on treatment options because they might work, which is what we have been left with for far too many years.
In closing, although we would do anything for another healthy minute to spend with Jason, we know that he is no longer suffering with clear cell sarcoma. But on behalf of our friends and family, we are certain that Jason's journey will make a difference. And we are honored to be here today and to take every step possible to help other patients, families, caregivers, and researchers to move forward. Our family and the Miller Memorial Fund, supported by our annual golf outing in Jason's memory, is excited to support the data collection project with hopes of making a huge difference. Thank you.
I'll never forget that day you called. And I'm so sorry. And thank you so much. We appreciate you. That's amazing. So those people waiting in the queue won't have to wait because we don't have the money. Sorry. Okay. Next, we're going to hear from the O'Briens, Donna and Kevin, who I have to give credit for connecting me how many years ago with Dr. Jones because of a post you put in the Clear Cell page in 2014. Donna wrote, "Hey, Dr. Jones is the only doctor working on clear cell sarcoma." And I read that and thought, and this was like maybe 2018, 2019. I said, well, no, because I would know about it. So I emailed Dr. Jones, or actually I called the office, and Diane, the amazing Diane, made sure that I got nowhere near you until she made sure I was okay. And I'm so thankful that we were able to connect, and thankful for the O'Briens. And I'm going to pass this on to hear about.
Thank you, Lenny. I probably stalked Dr. Jones too, but he would answer back. We emailed a lot. I mean, we text a lot, I think. Yeah, we text a lot. Kevin and Donna O'Brien, we're here to tell Cooper's story. We too had that terrible news about clear cell sarcoma, which was also misdiagnosed. Cooper was very active, and probably around 7 years old, he told me he had a knot under his arm, which, you know, young boys, as the pediatrician said, tend to have swollen lymph nodes when they're growing and that sort of thing. He tried to palpate it, but being underneath his arm, all Cooper did was laugh, and he kind of brushed it off. And of course, the next year, on his annual visit to the pediatrician, I said, well, he still has that knot, and and sometimes he complains about it. It's sore or it's hurting, and I just give him Advil or Tylenol or something, and it usually gets better, and he doesn't complain. So that went on until he got to be about 12, started playing baseball. He was pitching, and then all of a sudden, this monster started growing out of underneath his arm. So of course, we went to a surgeon, and he it was always said it was a enlarged lymph node or a reactive lymph node. So I said to Kevin, well, then we just need to get it taken out, because he's pitching and playing sports and it's going to always bother him. So let's just have it taken out. So we went to see a surgeon who just kind of reiterated, oh yes, reactive lymph nodes can enlarge and then they never really go back down. So we'll just take it out. Kevin had a trip he was going on. Cooper and I were going to the hospital just to have it removed. Simple as that. But, about 30 minutes after Cooper left to go under anesthesia for that surgery, which the surgeon now calls a biopsy, he comes back and says, well, it wasn't really a reactive lymph node. I said, well, what was it? He goes, not real sure, but probably lymphoma or sarcoma. Well, myself being in the medical profession, I know that's neither one are very good news. So I called Kevin. He had to turn around. And that's when our journey began, which was May 1st, 2013. We immediately went to see an oncologist at the Children's Hospital that handled solid tumors. And of course, the testing indicated melanoma, and Dr. Rapkin came in looking in his hair, looking on his body, looking for something that was a melanoma. And I'm like, I think I would know that if he had something that looked suspicious, because I'm just that kind of mother. And he decided to send it up to Michigan for the fusion test. Thankfully, I didn't have to beg for that to happen, but we were trying to figure out how could it be melanoma, but it came back clear cell sarcoma. So of course I was on the internet immediately researching, and we had that very serious conversation with Dr. Rapkin that, he liked to draw pictures and write things out, and basically told us it was a 50-50 chance of survival for 5 years. So we knew pretty early on, Kevin and I did, that we didn't share this with Cooper at the time, but he did know he had cancer. And in his mind, he visioned cancer patients as, chemo, and they've lost their hair. And he was like, no one's going to be my friend. And, I'm going to, lose my hair. And I'm like, I don't think so. But, once Dr. Rapkin told us that chemo was not, it's an option, but it had never proven to be successful, I said to Kevin, I will never make him do chemo unless I know 100% it's going to help. It's not going to be a what if. So we never had to do chemo because something to do. It would have just probably ended his life sooner. We decided to, number one, the surgeon, even though he was in there, he took a biopsy. He did not take all of the tumor. He left part of the tumor. So we had that to deal with. So we went to see a surgeon at Emory, and the plan was to have him to have radiation for 26 treatments and then have surgery to get the rest of the tumor out. And of course, after a diagnosis, we went for a PET, we went for imaging. And at that point, which was in June, he had no evidence of disease anywhere else. So the plan was to do the radiation. So we did that. And then we were scheduled for early August of 2013 to have surgery. But of course, it was also time to re-image. It'd been that 3-month mark. And of course, it was in his lungs at that time. So the surgery was canceled. The tumor was still in there. You know, at that point, there really wasn't anything to do. There was no options, you know, and that's the worst thing a parent can hear is that, you know, you want to do something, but there were no treatment protocol for this cancer. That was just starting in the market to have some targeted therapy drugs, not combos. There were no combos, just individual. So we tried 3 different targeted therapy drugs. I think I would send Dr. Jones like, well, what do you think about this? Or, you know, and it was really, I mean, we had a great oncologist. He would, whatever you want to do, Donna, you bring it to me, I'll make sure it happens. And he did. Those 3 drugs were very, very expensive. And I don't know how they got paid for. Worked for a hospital system and I'm sure meeting because we had a lot of expenses. But we were fortunate enough that we did get to drug. Pizza was probably the one that cleared up the lungs. But it was only probably a month and a half and some of those tumors came back, but it was promising and it made him feel better. Of course, you know, it does turn your hair white and your eyebrows white. And so he had his hair dyed and his eyebrows dyed. He's one of my friends is a hairdresser, so we took care of that. But you know, with Cooper's journey, I just remember anything about I found on the internet. If you just Google that, then their Texas came up, and we didn't have a Facebook page at the time. It was just communicating by email. And then Carolina was the one that actually built the Facebook page, and then I kept putting stories on there, and this is kind of how we, I guess, initially all connected. So I guess it was a good thing. And then mentioning Dr. Jones because he was kind of my hero. My brother-in-law found Mario Capecci's lab paper about a mouse model, a clear cell mouse model. And he contacted Mario Capecci, and who said, oh, well, you really need to talk to Dr. Jones. And so Michael, I think, got your communication, and that's kind of how we connected. And Dr. Jones sent me his book, and he was just so supportive. And, you know, I'm sure he got tired of like, who is this lady? But he was my only hope at that time. And we just continued to, you know, just enjoy every day we had with Cooper. And like you said, we just took one day at a time because we didn't know how long we would have with him. But it was 14 months and we did a lot. You know, we had a Make-A-Wish and we had the Falcon adventure, the Braves adventure, all kinds of things, the Hawks. I mean, they, you know, the community really, our small community of Swanee was, you know, wrapped their arms around us, and everyone wanted to do something for Cooper. You know, and somebody might show up and just bring a milkshake over and ring the doorbell and say, here's your milkshake, you know, and it was just truly amazing. But he did pass away on July 5th. His favorite holiday was July 4th. And it was just shortly after it turned July 5th that he passed away. We just, you know, the one thing I would say about the radiation, I do regret having that, but, you know, in the hopes of having the surgery and getting the tumor out, it was the best plan at the time, but side effects of the radiation ended up destroying his lymph nodes in his arm. And so he had a really hard time with lymphedema. So, you know, we found some lymphedema specialists, and they too were amazing, you know, but they're very rare. You know, trying to find one is very rare that specializes in, especially his arm, you know, was just grossly enlarged. So there were a lot of complications, and of course, you know, he went downhill pretty quickly, and it did spread all over his body. But for the most part, he was the best sick kid I think I've ever seen. He never complained. He went to school. He went through his 8th grade graduation. He was determined to do that. Anything else you want to add?
Great job.
Yeah. But my takeaway today, well, Kevin and I, you know, I think he probably more so than me wanted to just be done with it. But immediately afterwards, he was asked to speak at the Make-A-Wish fundraiser. I just don't know if I can do that. And I was like, well, but this is such a big opportunity. So how we kind of got thrown in advocacy is really by chance. Like I said, we both probably would have in the sand and just went on and just dealt with our grief. But of course, I wanted to, you know, find out why and help. And that was just kind of the path. We just kind of followed the light, and we formed a foundation in 2015, which is Cooper's Crew. And basically we, you know, try to help with research and inspiration to other people with cancer or any type of cancer really in our community. And we also do scholarships at his high school because he would have been a freshman at high school. And we do a scholarship each year for adversity and not academic at his high school. So we've been doing that since 2015. I think we've given away 42 scholarships and almost $100,000 worth of college help, you know, college scholarships. So we continue. It kind of takes a life of its own. It's not like we work hard and plan all these things. We just do our best to keep moving forward. But thank you.
I have to also say thank you. You've been very supportive to the Clear Cell Sarcoma Foundation, as well as the Sarcoma Foundation of America. So the O'Briens are very busy out there supporting sarcoma research, and the SFA is a good friend to the Clear Cell Sarcoma Foundation. We don't have a lot of opportunities, but we do have the Schaller Foundation is on as well, listening in, signed in. So there are, we call them our sister foundation for clear cell sarcoma across the pond over in the UK. But thank you guys for everything you do for us and for the sarcoma community. Next, I guess Anne is going to share her story with us.
Thank you. Okay. All right. So my name is Anne. Sorry, I'm getting a bit emotional hearing a 17-year-old had to pass away because of this, but I'm alive and I'm 40-something. So, okay. I was diagnosed with clear cell sarcoma in 2023. This is actually my first time talking about my journey into the world, so just bear with me. So I'm going to share my journey in 3 parts. If you can see from the slide, that's before I was diagnosed, during my diagnosis, and I wouldn't call it after because we all know there is no such thing as after clear cell sarcoma. I'm just going to say how I'm living right now in the present day. So back in November 2022, clear cell sarcoma were just pain and discomfort around my left ankle. I thought it was because I was sleeping in a weird position, so I just kind of brushed it off. After about a month or so, the pain continued, and I knew that I had to get seen for my ankle. So I made an appointment with a local podiatrist who diagnosed me with tendonitis. He told me to wear a brace and told me to follow up with him after a month. So during my follow-up, my ankle actually felt better with the brace. So we both thought that the brace was working. So I just continued wearing it. However, by March 2023, my ankle started swelling. You can see from the picture there was bruising. And I started noticing lumps around my ankle. And so I couldn't put weight on my foot at that point. So it was kind of like a foreshadow of what's going to happen to me afterwards. I was just hopping around with one leg at that point. My podiatrist saw me again, and he suspected tender tear and ordered an MRI. He saw a mass in the scan. He told me again and again it was very rare for ankle mass to be malignant. And he said that I should have surgery to remove it. And so I had no idea what was going on at that point, and I just kind of followed what he was saying. And I was waiting for the surgery date. But as I was waiting for him to give me the date, I started developing systemic symptoms. I had unexplained fever. I had shortness of breath. I felt dizzy. I started getting night sweats, and I was losing weight. Just even going to the bathroom to taking a shower was really, really hard. And at one point, I even became unconscious and passed out in the bathroom floor, which my husband was freaking out at that point. And so I spoke to the podiatrist again over the phone, and he told me to go to the ER and go see my primary physician. I took multiple trips to the ER, took multiple trips to my primary doctor. No one could tell me what the symptom was about, why I was having the symptom. My husband and I really thought that it was because of whatever was happening with my ankle and those masks that was found, but no doctor could take us seriously or even listen to us. And so one of the ER visit led to me having to stay in the hospital because they wanted to test me for Epstein-Barr virus, it's called. And it came back positive. And so, believe it or not, I actually felt relief because I had no idea why I kept getting fever and all that symptoms. And we really thought that was the reason why. And so my podiatrist said, as long as your symptoms get better, I can perform the surgery and take out that mass for you. And so I was really, really happy to hear that. And so again, back to waiting. Finally, he gave me a surgery date in April 2023. When I woke up from my surgery, my podiatrist had a really grim look on his face and told me that the situation was really bad. He saw extensive soft tissue degeneration around my ankle and a lot more things that it was just a blur. He was saying that it was just not what he was expecting. So he told me that he would leave my wound open because he needed to send a pathology report to get the result back. He said that if it was malignant, this is what they call staging, and that he would have the oncologist kind of deal with it afterwards to decide what happened. And if it's benign, he would go back into my ankle and try to repair possible tear in a tendon or anything of that sort. So you can see in the middle picture there, I have to kind of blur it out because it was really grotesque and I didn't want to shock anyone. But that was pretty much what happened. I was lying in bed with an open wound, waiting for the pathology report to come back. So I got the call on April 27, 2023, and he told me I was diagnosed with clear cell sarcoma. And so I had to do a lot of Googling because, like a lot of you, I did not know what sarcoma was, much less what clear cell sarcoma was. And my podiatrist told me he has to refer me to an oncologist. He just kept apologizing on the phone. And he said that, bad things happen to good people, and that was just bad luck for me. And so, yeah. So I went to see a medical oncologist, and when I was waiting for an appointment, I noticed that the tumor grew back in the same exact spot that the podiatrist did the surgery on. It grew back so quickly and so bad that it was surfacing out of my skin, and I could see it, which is why I kind of had to blur that picture because I didn't even want to look at it. But at the same time, I started noticing more mass around on the back of my calf. Also behind my knee. I didn't know what it was. I just thought because I was lying in bed for so long, I started getting some soreness or something. So when I finally saw the medical oncologist, he kind of recited what I found on Google, the same exact definition of clear cell sarcoma, and told me that I need to take some kind of chemo drug called carbomatrix. There was no suggestion about genomic testing or anything of that sort. He just sort of, that was like the first kind of suggestion he gave me. And so, you know, my family and I, we had no idea what we were dealing with. So of course, we were just like, sure, we'll try anything at this point. So they tried to get that approved with my insurance. And while I was waiting for my insurance to reply to me, my family said I should get a second opinion. So, Mom, if you're watching, thank you. So, you know, we decided to get a second opinion. And while I was waiting for the appointment with the second opinion, I heard back from my insurance, and the drug was denied because they consider it as experimental drug. And so, you know, there was nothing I could do at that point. The first oncologist that I spoke with said they're going to try to reach out again and try to get it approved. You know what, thinking back right now, I feel like it was all meant to be. I was not supposed to take that drug because I don't know what would happen if I did. But anyway, at the end of May 2023, I met with an orthopedic oncologist, which is the second opinion hospital that I reach out to. When he saw me, I just had a complete different feeling with this doctor. He had already read my records. He had read all of my scans. And he came in and gave me one possible treatment, which is amputation. He said, this is a very aggressive cancer, and so you need a very aggressive treatment plan. And at this point, because they found tumors along my groin in my lymph nodes, you know, amputation up to my hip will be the only option. And because there was also a nodule in my lung, he said if the nodule came back benign, we will proceed with the amputation. If the nodule came back malignant, you would have to talk to the medical oncologist to possibly look into systemic treatment. So thankfully, the nodule in my lung was benign. So I just keep remembering my family had a little celebration dance because we were so happy. And then coming back to reality, I had to decide whether or not I want this amputation. So my orthopedic surgeon was very, very respectful to what I want, but he said the only option at this point would really be amputation. So I went back to him again and again. Is there any other way? Because I really don't want to lose my leg. He said, if you wait, you know, I can't promise you there's not anything more I can do at that point. So it was a very, very difficult decision. But I finally decided to have amputation on June 9, 2023. And so anyway, when I woke up, it was really silly because the first thing I asked the nurse was like, do I still have both of my tushy left? And then my youngest later on would be like, Mommy, does that mean you have half a tushy now? And so, you know, I just kind of laugh it off, and it was really funny. Anyway, I did, you know, I proceeded with the amputation on June 9, 2023. Before I had the amputation, I was luckily able to find a Facebook group and talk to Lenny. And then Lenny reached out, and I'm thankful that she did. She said, you know, it's really hard for you right now, but would you consider donating your tissue? And so I said, you know, definitely, I would love to help any way I can. And she connected me to Barbara with pattern.org. And Barbara was really, really great with following up with me, emailing me, making sure that I got everything I need. And I consented, and I received the kit right before my surgery. And they also sent a kit to my orthopedic surgeon. And so it was really, really easy to do. And I'm glad I was able to do that after hearing all the updates and from Dr. Jones and Dr. Nobu. Because it seems like, you know, donating your tissue is really helping other people. And I wish no one would have to go through this like me. So anyway. So I was done with the surgery and back to present day. You can see in the picture, June 2023 to present. That was a picture, last picture I took of having both of my legs. So since my amputation, I've had quarterly MRI and CT scans to monitor to make sure that I'm doing okay. Thankfully, everything has been cleared. And after 1 year, I had a PET scan done to show that I have no evidence of recurring disease. And so I celebrated with a chocolate cake, which is my favorite. And so I've hit almost 14 months now with no evidence of disease and being an amputee for, again, over a year now. It was really hard being hit with a double whammy, right? You have to face cancer and I have to face amputation. I really miss my leg every day. But you know what? If I have to do it again and I have to choose again, I would choose my life over my limb any day. I'm really thankful to be alive. I'm thankful for my orthopedic surgeon. I don't know if he's watching. I don't think he knows about it, but I'm gonna send him this link afterwards. I'm sharing my story because I wanted to raise awareness about this ultra rare cancer. If you remember what Denise was saying, this is ultra rare. But I also want to highlight, no offense to Dr. Jones and Dr. Nobu, not all doctors are equipped to recognize the symptoms of sarcoma, especially ultra rare sarcoma. If you remember what my podiatrist was saying, he said he's never seen cancer around the ankle and he's never seen a malignant tumor around the ankle. And if he's watching today, I hope that I can be his first lesson that he will not do this to anyone else. Anyway, I am a former educator. I was a teacher for 15 years. And my biggest wish is that one day all the pre-med and medical program in the world will have a core subject on diagnosing sarcoma, at least recognizing the symptoms. As a matter of fact, I was able to find a study done by someone called Brandon Neal and some of his authors in 2019. The title of the study is called Student Perspectives on Oncology Curricula at United States Medical Schools. The study showed 4 key findings. Number 1, there's an underemphasis on cancer in the curriculum relative to other common diseases. Number 2, there's an imbalanced involvement of different clinical subspecialists as educators. Number 3, there was infrequent requirements for students to rotate through non-surgical oncologic clerkships, I think. And number 4, students are less confident in their knowledge of cancer treatment compared to basic science, natural history, or workup of diagnosis. Now, this study kind of gives an example of how oncology is underemphasized in the U.S. medical school curricula relative to cancer incidence in the population. I know that, you know, my little voice here will probably not make a difference. I just kind of hope that, you know, if my podiatrist would have referred me to someone that's more knowledgeable, maybe the outcome would have been a little bit different. Maybe my cancer wouldn't have spread so far along my leg. But you know, I just wanted to say that I hope that doctors in different kind of specialty, not just oncology, will be able to at least recognize some of the symptoms of sarcoma in the future after hearing so many people get misdiagnosed. But yeah, thank you for listening and, you know, bearing through all the sniffles. But yeah, okay. Yeah.
Okay, I first have to correct something. You're not a former teacher. You're still a teacher. You have all of us. She's an amazing teacher. You're also teaching the community about your experience. And you've just been, in my opinion, called to a new higher position. And we are so thankful that we have you and that you made the choices that you made, Ann, because and that you came. And at first was like, I'll just do it remotely. And we said, you're too important. And she made the trip. So thank you so much for that and for sharing about your experience. We have one more patient story from a family. I know everybody mentions here, Lenny called. I feel like I mean, I think what happens is we just, thank you, Donna, for bringing up Carolina, and because I found them too, the way you did before I found the page, and Kristen, who, rest her soul, was just recently passed away, and she was an amazing clear cell sarcoma warrior, fought for other patients. I called her the matriarch. She was the one. I have a pen with me that she gave me years ago. I carry everywhere. But there was quite a few of them in the beginning. I think I shared with Denise. I think this is important for me to share, talk after all these patient stories and hearing. Do you remember the document I brought you that I said I felt like you were the right person to give it to? So you guys, it's still in our Facebook page. I don't know if anybody pays attention, but they actually did the first ever clear cell sarcoma patient registry on their own, color coded by each patient. And they passed it around. Carolina and the group, Mike, the Gratz family, who is still amazing themselves. They're another, still helping other patients. But these guys, there was maybe 10 or 15 of them. And they wrote, did you have an injury there? Where was it? Did you were you misdiagnosed? What treatments did you take? This group of patients who had nowhere else to turn, there was no foundation. And I even saw the original text where they had to be secret about sharing their names on that. There was some site, like a patient reporting site, and it was Carolina was Caro. And I was like, who's the MG? And it was them. And they and they were so amazing. I have to share this. They deserve this minute. There was a Chinese clear cell sarcoma patient who I guess wasn't allowed to have a Facebook account. So this small group had an email for him, and they would share all their discussions across email. Carolina was in charge of it. And that's how committed, like, if you can imagine the whole world in a time before everybody, it was 2009. When was Facebook even created? It was probably right in that time. And they were gathering in just unbelievable circumstances to do that. So I can only do the work I do, and the foundation's been as successful as it's been because of what was started there. You would agree. The O'Briens have been around for a long time. When I joined in 2018, there was only 68 members of that page, and it had been around since 2000 for 8 years at that time. And there was 68 members. We have over 300 now. Right. Me too. Exactly. And I remember every one of us is going to remember the first other family that we spoke to with clear cell. It doesn't matter where they were in the world. And I remember Sandy Staffieri, who I hope she's logged in. I know she'll listen, but for hours, let me walk across the back of the field at my daughter's softball game and just unload. And that's what we do and what we're here for. But we do want to give them that minute, because there are many that came before us, and we're here today and able to do what we do because of them. So Brian, his daughter, we're going to play his video. His daughter, Erica, when you do talk to somebody, you're reliving it in some ways with them. And then you're just people did it for me. You're trying to pour out onto them everything to get them those miles ahead, that maybe it'll be a little bit different, or they'll get that second opinion a little faster. And so Erica was very special, and She did end up with an amputation as well. She had it in her hand and it was a kind of a botched surgery. This same story. And she had 2 small children and wanted to live for her children. And she went ahead with the amputation. And I think she was able to buy some time with it, just several years. But her father is committed to sharing her experience here. And something I want to point out that I think is important when I watched this video, because I've heard this story a lot. So this is for the clinicians and the scientists. The pain in the end caused by the sarcomas growing is not understood. And I will even say when my daughter had her recurrence, we made multiple visits to the ER, and every single time I would tell them, we've been doing this. Dilaudid is the only thing that touches it, but they would still make her go through morphine, oxy. We had to go through while she just writhed in pain. And I didn't know then what I know now. And Erica's story is very similar. And I hope that we can get this information out there too in the data, because patients, they just don't need to suffer in the pain while a doctor tries to think, I don't know what they're thinking, if they're faking it or but they've said that you can't be in that much pain or this has to be working. And it's really, that's the hardest thing. I mean, I won't go into my story because mine was praying to God, if this wasn't going to work out, take her fast, because she was in so much pain. So that's just something I want to make sure we also capture in the data about, because I've had to hear that story over and over. And we'll go ahead now and play Brian's video.
Brian, you're in the Zoom. So just let us know if you want to say a few words about the same video. Can you hear? Okay.
Jason, can you hear it? No. Okay. We'll get it working. Just bear with us. Okay, should we play it on a different computer? So we
Let
have
me-
this first. Okay.
Let me try again. Can you hear anything? No?
While you flow.
Oh, yeah. Yeah, it's back.
The microphone.
Can you try it? Can you hear?
Do you want me to answer it? Okay. Oh, I'm sorry. So.
Can you hear?
No, boo. In fact, I'm hoping that your work is going to help us make a better argument for getting it back. We completed.
Oh, but I switched it back to the one before.
Phase 1, early 23. No, it's been a while. Yeah, early 23. All safety measures were enhanced. Jill and I, well, there's a lot more to it, but the NCI, the National Cancer Institute, had indicated they'd be interested in picking up
No.
the phase 2 portion if the pharmaceutical company paid for
No.
the phase 1, which we funded the travel for the patients. We passed all safety studies, but are we good? And the answer to us was, Well, we didn't see efficacy. Funny that they said that, because when I was involved in setting up the trial, and we talked about, they were letting, it's a phase 1. So a lot of the patients that go in a phase 1 are told hospice or phase 1, and everybody wants that Hail Mary shot. And what I was told when I said, well, even the evidence from the Japanese paper that you referenced shows that this works best in early phase and low tumor burden. And what they looked at us and said that, and this came from our friends at the FDA, they said, don't worry about it. We don't look at efficacy in phase 1, not even going to look at it. Well, when the phase 1 was finished, they wanted to see efficacy before they took on the phase 2, right? Dr. Jones smiles. Yes, that's how things go. So we do have some plans in the works. We have been speaking with the amazing CEO that helped us get it. We can't say enough thanks to Raphael, now Cornerstone Pharmaceuticals, for doing that phase 1. We are not going to stop trying, and Dr. Nobu's presentation just gives us more ammunition. And so, yeah. So do we think we have Brian's story? There we go.
Yeah, Erica was diagnosed with CCS 2019, and she fought for a year with treatments, with radiation, chemo. Actually, I think she just might have had radiation, and she had direct radiation. So her CCS was on her right hand, on the palm at her thumb. And so they did direct radiation to that spot for quite a while. She went through several treatments, and it did no good. So after a year, they decided to amputate her right hand, which she went to Cleveland Clinic and had that procedure done. All in all, I'll say Erica did better than I probably would have with it. You know, she got isolated there for a bit, was embarrassed because of her hand being gone. She had a hard time because she was right-handed. Good morning. My name is Brian Gould. I am making.
Who's doing that? I was just. Oh, no, sorry. Is somebody else controlling it? That's what I was saying. I'm just saying you can turn up here at top. Sorry, we have. It wouldn't be a good Zoom if we didn't have some. Sure. We have another question. Because Brian's story is very important. Another question. Yeah, you got the questions up, the originals?
My name is better than I probably would have with it. She got my there for a bit, was embarrassed because of her hand being gone. She had a hard time because she was right-handed, so it was her right hand that got taken. So her primary hand was gone. Erica has 3 children, and it was very hard for her to get her girls' hairs done, get them ready for school, get herself ready. It was very difficult on her. And then 3 years after her diagnosis, she had scans done and it had went to her lungs before the amputation. So we were in another fight. Once again, she went through radiation for that. Ended up having a spot on her larynx, and one right behind her heart on her spine. And Erica ended up losing her life on June 26th of 2023. And part of the reason I want to make this video is, yes, to help with the diagnosis and the treatment and the care for the patient and the family during that process. But the absolute worst for the family was Erica had been admitted to the hospital with chronic back pains. And the whole time that she was there, they knew she wasn't going home, and the pain was caused from the growth of the tumors, not just regular back pain. And we did not know that for 6 days while we were in the hospital. And we found out some things about Erica during that time. And so I approached her doctor at St. Vincent's here in Indianapolis about the situations that we have found out about. And that's when she informed me that Erica was not going to go home and she only had a few days to live. And nobody had told Erica or the family this yet. And not only that, but I had to be the one to sit with my daughter and explain to her that she was not going home and that she was only going to be with us for a few more days. So, you know, that's really my thing right now is just how do we educate the caregivers? And no, by no means are we blaming anybody for this. She was the first case in this hospital. And so, you know, how do we get these first cases to not have to go through what we went through? And I would love to tell you more about the hospital stay because there's a lot to it, and I don't want to take up too much of your time with it today. But you know, the hospice care, just the overall stay. She was in the ICU for several days, and they, you know, we had some real issues there because they didn't understand what she was going through. And so they treated her like a regular patient, not somebody who was only going to be alive for a few more days. And so her last week was not what it should have been or could have been. And it definitely wasn't what we were wanting it to be for her. So, yeah, that, you know, like I said, I care all the way from step one to the end, but the end for us was absolutely horrible. And I don't want to see another human being ever have to go through something like that. So, yeah, that's kind of our story. And like I said, I'd love to talk more about it if you guys are interested or need to hear or want to hear. I can go into detail. It's a lot of detail. So, like I said, I didn't want to go into it with you today, but I'd be happy to do it at some other time. So, once again, I'm doing this for my daughter, Erica Jane Gould. She died June 26th of 2023. And I really hope that Lenny and the team can really start making some good strides with helping the patients and the families through the process. So anything I can do, I'm here and I'll do it. So I appreciate your time and hope you guys have a good day. Thank you.
And that you can ask him, sure. So, yeah, Brian has our clear cell sarcoma family, that a lot of you here know him, but I mean, I get the calls all the time. What can I do? What can we do? How can we help somebody? If you go look at our mission statement, it's been the same since the foundation began, because it's something that was really important to me, is that if we were going to do this and I was going to commit my life to this, it had to be a place where patients would be able to come, because we felt the same way. Like nobody knew anything to tell us. They knew there was things they wanted to do to my daughter, but there was no, you know, I guess gathering of information, gathering of something to make a difference. So we wanted to do that. Trust me, we called the SFA, we called everybody. Do we really need to form a foundation? And you do. You need to have your community in an organized manner. But when we did, we came up with our mission statement. It is to give the patients a platform to make change and strive toward the cure. So that's what is happening here. And I'm pretty excited that we've been able to come together and hold this symposium. And I'm so thankful when I called, and I got to say, we didn't ask, it was important to us that we asked true clear cell sarcoma champions. And our first one to call was Dr. Jones, and he, like he has done in the past, immediately said yes. And Denise helped me kind of frame it. I mean, I took it to Denise first. Here's my idea. Here's what you need to do. And Barbara, these are true champions for this disease. And then, of course, all the families and xCures and who we're working with. But we hope to grow it. There's a lot of doctors out there who are amazing. And I know they're doing good work on clear cell in other countries. It's really sometimes hard. But I hope that the next symposium, we double our size and we have more. And I think like a snowball, it's going to get done and we're going to get there. Now, we did go over a little bit. We've got a little bit of time. If we don't get to your question, we will answer it in an email to you when we send the, right, Ann, am I saying it right? When we send everybody the copy of the recording, we'll also get some answers to their questions. But we do have a few minutes. Yeah, we actually do have a little bit of time before lunch. I've got them up if I need to go back to my computer. Okay.
Oh, I'll read it. Okay. So the first one is for Denise. How do I fight against the insurance refusing to pay for treatment for CCS because it is not approved treatment until I fail traditional chemotherapy that is also unapproved?
That is a great question. And I'll share my thoughts, but then I probably am going to pass the mic to Dr. Jones because I would like to hear also his perspective on this. I think this is, there are two conundrums here. One, there is no real approved systemic therapy. And so if you were to really, even for what we call a traditional chemotherapy, that's not, systemic chemotherapy is not approved by the FDA. So when we're talking about FDA, and oftentimes insurance companies will use what's kind of listed as approved. We talked a little bit about guidelines and national guidelines, and one of the reasons I think we've kind of lighted upon the National Comprehensive Cancer Network guideline, because many insurance companies will utilize those guidelines as evidence to pay. So if it's written in the guidelines as a treatment, it would be approved to be paid for by insurance if it's given. And those guidelines, just by background, are consensus-driven guidelines where expert panels of people with sarcoma expertise review the literature, what's published out there. Of course, the highest level of evidence is doing a high-level phase 3 placebo-controlled study is the highest level of evidence. And in many of the subtypes of sarcoma, we cannot do that kind of research. So what's really unique about these NCCN panels is that they seek to come to consensus about the data that is published in the literature. And so they really are synthesizing all that information in the guidelines. And so I think that, and the reason I'm spending time on this is how important it is as an opportunity to get the data that's needed so that the expert panel can review it and say, is this sufficient evidence that we can stand behind it, put it to the guidelines, and hopefully help to pay for it? I think the other part of this is that because we don't have approved treatments, it really makes it so critically important that we get clinical research out there so that, and twofold. One is that when we're giving new therapies that we really don't know for sure, that maybe it's based on strong laboratory findings and evidence that we think this is going to work, that we do it in a way that we're capturing all the data in as precise a way as we can, so that we can amass that evidence, and then insurance and payment won't be the obstacle for people to get access to the care.
Do you want to add to that? Do you have anything to add? Sure. Okay. I know these are tough questions because.
Yeah, so I mean, I think many of the questions on here relate to this principal question though. And I think there are proven things, there are unproven things, and then there are possible things. And these are all very different for a rare disease like clear cell. So a lot of the things that people are doing are things that are FDA approved from the standpoint that they have been FDA approved for some other cancer, for some sarcoma, or have been put forth as a protocol for a trial, usually by most of the truly prospective trials that have been done in sarcoma chemotherapy have been done in children. I mean, most of them were done by the Children's Oncology Group. SARC has done a bunch. But even most of theirs are not truly placebo-controlled, trials. And so a lot of things, if something is approved by the FDA for anything, a physician can usually order it for a patient. Sometimes they can get it approved by insurance, but that's not always. And insurance companies these days push back more and more on these things. And they push back on them, especially if they are newer treatments. I mean, it's not like newer treatments, targeted therapies are in the whole a lot more expensive than traditional chemotherapy because they usually don't involve so many hospital stays. They don't, involve ICU care for the complications as often and things like that. But they still, it is a little bit easier to get doxorubicin approved at, I don't know what a dose of doxorubicin costs, but I'm guessing it's going to be $1,000 or $2 per dose, compared to a targeted therapy, which is, an immunotherapy or pazopanib or one of these types of targeted therapies, that it's going to be tens of thousands of dollars per month. And so a lot of insurance companies are going to balk at those, especially if there's not proven efficacy for any sarcoma, let alone a specific sarcoma, which is why we hear most of these patients, if they've gotten a targeted therapy, it has been pazopanib because that is approved for sarcoma writ large. It hasn't really been tested in a serious way in clear cell sarcoma, but it has been tested for sarcoma as a part of its FDA designation. And so that a lot of these these things come down to regulations, and sometimes the regulations are in our way. Sometimes they are also trying to keep physicians from kind of being mercenary or predatory in their practices, where they're offering things that have no real promise. Again, the big challenge with clear cell sarcoma is that nothing has great promise other than maybe if you're able to cut it out. And sometimes those are very drastic things, as Anne experienced. I think when it comes to a rare cancer where we don't have proven therapies, to some extent, I tell my kind of anything goes. I mean, you can treat this as a general sarcoma, applying general sarcoma principles, which is where you're going to get to radiation. You're going to get to when you have systemic disease. We're going to try things like doxorubicin and ifosfamide, or Ewing sarcoma, chemotherapy, because Ewing sarcoma is a little bit molecularly related to clear cell sarcoma, which adds to doxorubicin and ifosfamide a couple of other drugs. But because nothing is known, anything goes. But I always tell my patients as well with that, be careful that anything you try, follows the reasonable test. If it's going to make you broke or miserable, I don't know that I'd do it. And part of the problem is that a lot of our traditional treatments for sarcoma do make people both broke and miserable. And that's a serious challenge. Usually they are covered by insurance, and so they make your insurance broke and just you miserable. But these, when we're in a rare disease space, an ultra rare disease space, all of this has to be really sort of carefully tailored within whatever guidelines can get it paid for, and within whatever guidelines are appropriate for state licensure and all those kinds of things. I mean, there are other issues that can come to play, but that's why I think at this point it mostly requires really good communication between your physician and the patient and family, and ideally keeping track so that we know, or at least we're gathering information so that 10 years from now we're not in the same position.
Thank you. Yeah, Denise wants to add something. And while I'm walking over there, I'll say I've had some experience helping patients walk through insurance issues. And one thing I can, for any patient or families listening, I highly recommend in the US, if you are with any of the big insurance companies, call your insurance, call the number on the back of the card, ask for a nurse case manager, get that person assigned to you. Almost all insurance companies now have some kind of concierge service, but get a nurse case manager assigned to you. Then let that person know what's going on. Teach them about clear cell. They will fight for you. They will fight. You can even go to the NCI website and look up clear cell or sarcoma. Go to the Sarcoma Foundation of America. Look up this information. There's documentation from these organizations that you need to be in a high volume sarcoma center. We were able to get full payment for my daughter out of network. We were actually able to get, and I had someone helping me with this. We were able to get Blue Cross Blue Shield of South Carolina to put Memorial Sloan Kettering in network for an N equals 1, I guess. And so don't give up. There are ways to do it. Use logic. Use logic and think, okay, like the question asked, why are you going to prescribe one unapproved drug before I can get another unapproved drug? Use the logic that this is a very ultra rare, and then also it might cost you more money to not, let me try this or go to a high volume center and see what they're prescribing. So sorry, but that's.
Yeah, no, but it's a lot of energy, and at a time when you are facing so many struggles. And I think to Dr. Jones's last point about capturing the information, I think, the FDA has a responsibility as a regulator to set certain rules so only things that are safe are approved. And then in this whole approval process, it gets to be a little bit of a quagmire of if it's sarcoma in general or a specific subtype. And is your insurance smart enough to know that clear cell sarcoma is different than? And so I think there's such variability across different insurance companies. But I think the initiative of the FDA through the Cure ID program, they are trying to understand and capture the data just that you're talking about, Dr. Jones, so that we can understand what have you used, and can they identify something that might be approved in another indication that has been shown to be effective in a rare cancer. And now they're looking specifically at sarcomas to say, are there things that people are doing? Because we know those randomized high-level evidence studies cannot be done in these rare tumor types. And so I feel like that's just like almost always it seemed like that, it's like this big government kind of official place that you couldn't ever knock on their door. I think they're opening the door, recognizing that there's a problem and we need to try to, and data and information, and Hannah spoke to loving data and how important data is. It really is. And so I think this whole initiative of capturing the information so that 10 years from now, we're not saying, people tried all these different things, but we didn't co-locate that information in one spot so that we can say, this is helpful or this is not helpful, or maybe there's something already approved that could, and I think that they haven't directly said this, but the reason that the Oncology Center of Excellence of the FDA is supporting this is to see, is this potentially a pathway to expand some labeling, because companies are not going to spend money to do jump through all the hoops that they need to for an approval of a very tiny population, because at the end of the day, they're for-profit companies. And, then sometimes it just doesn't make economic sense, which is also another horrible part of this whole situation. So sorry, kind of a long-winded answer, but
True. And I agree with you. And I have to say, Heather and those at the FDA that we've been working with, they are trying to gather this data and they're very interested in the project we're doing with xCures and we are very interested in sharing it so that we can continue to help others, because it's going to take all of us bringing those connecting those dots to make a change. Because we've seen decades now, as you even showed us when you brought that one paper up. I've mentioned many times that Enzinger paper from the 60s. I'm like, wow, this reads like, the only reason I know it wasn't written yesterday is because it was on a typewriter. So we thank y'all both. We have another question, Anne. Okay, yeah, here's one for xCures. With the xCures platform, does it have the capability to identify trends, patterns, cross-reference my records with others similar to my case, and then use AI to recommend next steps? For example, if my scans consistently show a nodule in my lungs and there are others like me, I'd like xCures to show me what treatments, procedures these other patients have tried so I can share it with my oncologist. Okay, that's part of our partnership. Can I take that?
That's a great question. Unfortunately, no, at this time, we do not have that analytics layer. However, that is one of the reasons that xCures is in place, because to be able to do that, you need that robust data. And that is what our focus is on, is gathering that robust data to be able to share with companies that do that, so that they have accurate, clean, robust data to be able to help patients and provide that kind of treatment options that they're looking at. Yeah, that's it. So sure.
As we gather the clear cell sarcoma data from the patients, if there's patterns across the CCS patients, that's the kind of stuff we look for too, right? So technically, yes, as an individual organization working with y'all, if we're looking for that across our patient population, which hopefully then would transfer across the sarcoma patient population. So there's hope for that.
And I think you mentioned before something with Sarah's previous labs and indicator that you had looked at before. So that's what Hannah was speaking on, is to, yes, we do have this clean data, but there's also real world data that can identify trends prior to diagnosis that would help someone else.
And every day, as we develop our platform and incorporate more features available through AI, I mean, the growth and possibilities are exponential at this point. So what we're pulling back and extracting automatically today with labs, vitals, conditions, it's becoming more and more robust and giving us more possibilities in cleaning the data and being able to perform analysis on the data set. And so with that, right now it's as we mentioned, we don't do a lot of specific verification on labs and vitals because the accuracy of what we're pulling back automatically is so high. So we're working every day to get to that point consistently with our procedures and our conditions, et cetera. So what we're doing for CCSF right now is going in and using human resources to make sure all of that is verified and accurate. But as the future progresses, I do see a world where you're asking, okay, who has had this condition and what was the treatment and what was the outcome? Right now, we're in ultra rare disease, so the N is very small there, but the more data that we can get around this and the more patients that participate, the more robust and powerful the data outcomes will be.
You can read it.
Yes, I can. This is a question I get a lot. For xCures, what will happen to my medical records if I decide to stop using the platform? Also, can I request xCures to send all of my medical records from different facilities to somewhere else? So if at any point you can opt out, we just need it in, legally in writing saying that you want to opt out from using the platform and from us continuing to collect your records. I always like to tell patients that and loved ones that everything is de-identified and sometimes we'll just refer to it as scrubbed, meaning that all patient identifiers are removed. So you can have that peace of mind that even if you do want to opt out, everything that has been collected is scrubbed, meaning that there's nothing that could point it back to you, even with using timelines for dates and treatments and that sort of thing. To this time, we do not send any records to other facilities like you would request from an institution. Someone couldn't request us to send all of their records somewhere else, but you could go into the platform. And one of the things that we're working on currently, you go into the platform and you're able to download the records that we've pulled from the Health Information Exchange. So you can go in and I could show an example later if we had time of what that looks like, what the platform looks like, a demo of that. So you can go in and look and see everything that's pulled electronically or anything that we've also received via manual fax, and you can go in and upload it yourself. So our goal is, obviously to have all of the medical records for a patient in one place so that you can easily take that with you when you go to another institution, et cetera. So I hope that answers your questions. Yes.
Thank you. I'll say too, the four of us started with Sarah and Hannah as a practice group so we could see what it was like so we would understand the process. And I think that was really, really helpful because we could see our own data, my daughter's data, Lisa, and then Jill's daughter's data, that our daughters consented. And we were able to look at it and then look at where maybe the gaps were, where the needs were, maybe. So it really, and this is the beauty of being able to work directly with xCures and them willing to work as a partnership collaboration with us, because it helps them to build a better platform for us, because they don't know clear cell sarcoma, just like we sit here and talk about all of our doctors might see it once. Can you imagine? So even when I always think when you say the de-identified, yeah, it is de-identified, but then we've got Kim and others over here who know, well, I've been talking on the phone with Lennie for hours and she's asking me these questions, because when we get the data, that's why we've started with such a small group. We know your family story because you've told it to us and you've contacted us that you want to be a part of this. And when we get, say there's 4 of us and then we add 2 more, there's 6, we'll get it in a pie chart. And the pie chart numbers won't look right for what we know about those 6 people. So we go back to xCures and then we say, okay. And then we go back to the family. And so, it's not going to be a requirement for those of you who are consenting that you're that active, but the more we're starting with the active families, because the more we teach, am I saying this right, y'all? The more we teach the AI what we're looking for, the better it's going to come down the road. And Hannah has been teaching us about normalizing data and what that means. And I'm going to also tell everybody out there, saying that manual manipulation does not mean we're changing data. I thought so. I was very much against that. Manual, what is it that you told me? Manual manipulation just means you need human intervention. Normalization.
So the data that we get through real world data is messy. It's everything you can possibly imagine, buried in clinic notes, buried in different kinds of records, fax records. It's messy. And so what we're working to do is extract that data in a normalized fashion. And once the data has been normalized, in order to analyze it, often what you hear in the data world is the word manipulation. We need to manipulate the data to understand what it's telling us. So manipulation, though, has a negative connotation in the world of data, is very common, and something that actually is important. We could probably come up with a better term, though.
For us lay people, so I'm sitting in between like what I think. Also, normalization was a word I didn't understand until you told me. I'm going to give the example that we actually found: excision, surgery, two words that can mean the same thing coming from different institutions. So you would want that if someone had surgery, one person had excision, one person had surgery, you'd want that those terms normalized. So we're getting accurate, complete, good, and that's when Hannah says clean data. So do we have it? Okay, we have a few minutes. Okay. Do you want to read it?
Yeah. The first one says, I'm Ned, no evidence of disease, and my surgery was over a year ago. I asked my oncologist about adjuvant therapy. I was told that I don't qualify because he said that it's for high-risk patients for which they have data that adjuvant therapy improves outcome, and the time frame to start the therapy is 4 to 6 weeks after surgery. Is there any other preventative protocol that I can try?
Yeah, I would say adjuvant therapy is a shot in the dark for clear cell sarcoma of any kind. I mean, we just don't know, and so because of that, you're going to be subjected to whatever your physician's opinions are. You know, so I can tell you, because I'm a surgeon at both an adult cancer center and a pediatric cancer center, those are very different. So like pediatric oncologists or the chemotherapy doctors who are at our children's hospital are very aggressive. I mean, they would not ask permission. Like they would just, that every patient with clear cell is getting Ewing sarcoma chemotherapy, because like that's just what they do for that. On the adult side, where they're used to treating a lot of sarcomas that don't respond so well to chemotherapy, as most soft tissue sarcomas don't, they're a lot more sort of circumspect about it, and they would usually say, let's have a discussion. And in our center, mostly because of my strong opinion, but they would not typically treat soft tissue sarcomas with any chemotherapy unless they had metastatic disease, and then it's a discussion. Then we know we're in a shot in the dark kind of situation. So I think you are subject to the opinion of whatever oncologist you see, and in these areas that are sort of data-free zones, they're very much going to put you into a bin, different bins. And if they put you into the general soft tissue sarcoma bin, they're likely to, depending on where you are, if you're at MD Anderson or you're maybe at Sloan Kettering, they're going to talk about systemic chemotherapy. If you're most other places in the country at sarcoma centers, they will probably be slow to do chemotherapy for a soft tissue sarcoma. But if you're in a pediatric center where they're used to rhabdomyosarcoma, very different kind of sarcoma, but they all respond to chemotherapy, at least initially. And so that is their sort of, you know, paradigm for treating it, and you're going to be subject to that.
Good. Here, I'm looking, someone asked this question, and I think you're perfect. Should a pediatric or young adult sarcoma patient receive a different treatment than an adult patient? I'm interested.
In my opinion, no. But again, that's my opinion. There are other people who are entitled to opinions. none of us has any data. And so that is the challenge in a rare disease space. There is some data. There is some data. But I would say that none of that is care directing data. We don't have any level 1 comparison of drug A to drug B to placebo or whatever for a large cohort of clear cell sarcoma patients ever.
Well, started. And I think it's a great way to end this part of the day because it's now started and we're going to have the rest of the days is not going to be on the webinar. It's going to be a closed discussion, but about exactly that. So
yes. Yeah. So let me just put a plug for that. Because we don't have any what they call in medicine level 1 data, we have to get the very best data from real world data, as they call it sometimes, or empiric data, because that's all we have. And that's all we're going to have in any reasonable time, unless we discover something that is, miraculous level efficacy, which if we do, then it will likely, proceed to real, clinical trials in a very fast way. That right now is still a pipe dream. And so in the absence of that, what we really need to know is how the different things that are being tried are working out. And so the best way to know that is for people to register their data, and including all of their data. One important thing about the whole xCures platform is that they are pulling medical data. You need to tell your doctors if you're doing other things. If you have a special diet, talk to your doctors about it so they can record it in the medical record and xCures can find it. If you're keeping that from your doctor because you're afraid they're going to think badly of it, which maybe they will, depending on your doctor, but hopefully they won't. But they will at least record it because we have to know those things too. And if you're trying some supplements, if you're trying some, other medicine that is not a cancer medicine that sometimes are being used for cancer, all these things need to be recorded so that we can begin to understand as we get decent quality data collected, we can begin to at least to start asking the right questions. Thanks.
okay, so
Thank you everyone for joining us. I hope this was informational and helpful. The recording will be posted on Clear Cell Sarcoma website and also on our Facebook page. It will also be emailed to everyone who registered. We will also put together the question along with the responses from the speaker on our website and also on Facebook page as well. And so if you have any other question, please feel free to use that Google form link that was sent out and continue to submit your question. Again, thank you for joining us. We hope you have a wonderful rest of your day.